Hello everyone, the Nerd Medic here and you're watching my YouTube channel. In this video we will learn about the concept of erythropoiesis. Now over here the term erythro simply refers to the erythrocytes, right, which are essentially our RBCs. And how these RBCs are synthesized, that process is known as poiesis. So it is the process of synthesis of RBCs. Now we will see in this video how the RBCs are produced. So for that we have to understand a basic story. Now what is the basic story? The story starts at around third week of intrauterine life. Right, in third week of intrauterine life, within the fetus or embryo, we have a place known as the yolk sac. Now in the yolk sac, hematopoietic stem cells appear. Hematopoietic stem cells, these hematopoietic stem cells ultimately give rise to the different blood cells, right? Now at around third week in the yolk sac, the hematopoietic stem cells appear. Now these hematopoietic stem cells at around third month of intrauterine life, they move mainly to liver, and to some extent few of them move to spleen and lymph node. Right. So at around third month, as the yolk sac is doing a bit of hematopoiesis, but most of the hematopoiesis is being done by liver, with some portion being done in spleen and lymph node. This hematopoietic stem cells at around the time of fourth month of intrauterine life move to somewhere which is known as the bone marrow, right? And from this point onwards, bone marrow becomes the major site of hematopoiesis, even though during the entire fetal period, liver, lymph node, spleen, to some extent yolk sac, along with the main site bone marrow, keep on doing hematopoiesis. Erythropoiesis is a part of hematopoiesis. Hematopoiesis means synthesis of blood cells. Erythrocyte or RBC is a blood cell. So erythropoiesis is nothing but a subpart or subdivision of hematopoiesis. Now this scenario changes at around birth. So at birth, bone marrow becomes the major site of hematopoiesis. Right? Bone marrow becomes the major site of hematopoiesis. So this is the story before birth, what happens in the different organs regarding hematopoiesis or erythropoiesis. Now let us see what is bone marrow, where bone marrow resides. For that we have to see a bone. Now if you take a random long bone, most of the long bones will have this kind of a structure. If you take a cross section, you will see the long bones have two parts. The outer part is a solid tire of bone, as you can see. So this is known as the cortical bone. And the inner part, you can see that inner part is very much meshy. It has a network like structure and there are many holes are punched out. So this structure is known as medullary bone. Over here, remember one concept always, that whenever you have a tissue, the outer part of the tissue is always known as cortex and the inner part is known as the medulla. So similarly, in case of a bone also, the outer part is known as cortex and the inner part is known as medulla. So the inner part of the bone is known as medullary bone and the outer part of the bone is known as cortical bone, right? Now the inner part of the bone, there are many holes punched out. So this gives an appearance of a sponge. So this is known as a spongy bone, right? And also there are so many parts cancelled out. So it is also known as a cancellous bone. We will talk about these things in details when we will be discussing about the anatomy of a bone. But right now we have to understand that the bone marrow that we were talking about so long, that resides in the medullary cavity. Right? And this bone marrow is responsible for the hematopoiesis and for our discussion, the erythropoiesis. Right? So now let us get into this cortical bone, cancellous bone or the medullary cavity or the bone marrow, so that we can visualize the erythropoiesis process. Now in the cancellous bone, we know there are certain blood capillaries. Now these blood capillaries are very special. These blood capillaries are known as sinusoids. What are the sinusoids? Sinusoids are special blood capillaries which have distinct spaces in between the endothelial cells. Even though in this diagram the spaces are not very much distinct, but remember that sinusoids are special type of blood capillaries where you have spaces between the endothelial cells. Through those spaces, the cells which are produced in the bone marrow get into the circulation. So remember very well. Now over here we will see one cell which is known as the hematopoietic stem cell. Now this is the very beginning of the process of hematopoiesis or erythropoiesis, right? So this is a stem cell, and remember that this is a pluripotent stem cell, right? It's a pluripotent stem cell. Now this potency of a cell is determined by which type of cells a particular stem cell can produce. Now potency is a whole different concept which we will take up in a separate video, but for now just remember that hematopoietic stem cell is a pluripotent stem cell. Now from this hematopoietic stem cell we will get something known as the common myeloid progenitor, which is a multipotent stem cell. So its potency is a bit less. So multipotency is lesser than pluripotency. So it becomes more specialized. As you lose your potency, you become more and more specialized or goal directed. Now from this common myeloid progenitor, you will have something known as megakaryoid erythroid progenitor. So it becomes more specialized. Now the hematopoietic stem cell could have given rise to all sort of blood cells. Then it gave rise to one kind of cell which is known as common myeloid progenitor. So that become a cell which can only give rise to myeloid progenitors or myeloid cells. Then it again got more specialized. So it became megakaryoid and erythroid progenitor. That means it can ultimately give rise to megakaryocyte and erythrocyte. Now from this erythroid progenitor, we come to something known as the proerythroblast. So this proerythroblast is also known as pronormoblast, because just after this whatever we get is known as the early normoblast. Now after early normoblast we get something known as the intermediate normoblast, and after intermediate normoblast we get something known as the late normoblast, and from the late normoblast we get something known as reticulocyte, and the reticulocyte ultimately gets converted into RBC. So let us summarize the process once. So from hematopoietic stem cell to common myeloid progenitor, from common myeloid progenitor to megakaryoid erythroid progenitor, from that we are coming to proerythroblast, which is also known as pronormoblast, because after that we get a series of normoblasts. Then we get early normoblast, intermediate normoblast, late normoblast, then reticulocyte, and finally RBC. Now let us delve even deeper into this. See, first of all, hematopoietic stem cell has few features. Over here it has a large nucleus, right? It has evident euchromatins and small basophilic cytoplasm. So this large nucleus, euchromatin, basophilic cytoplasms are all features of a metabolically active cell. A metabolically active cell requires to produce a lot of gene expression. So for that you have a large nucleus. Most of the genome or genetic material is in euchromatin format because a lot of transcription is happening. And why basophilic cytoplasm? The transcript, that is the RNA is coming into the cytoplasm. There's a lot of RNA, lot of ribosomes in the cytoplasm which are going to produce the proteins. And as RNAs are acid, they are basophilic, they love bases. So they bind to basic dye, and that's why they give us basophilic appearance to a cytoplasm. So always remember, whenever you have a metabolically active cell, these features will be seen. What is this triad of features? Large nucleus, euchromatin, and basophilic cytoplasm. So these same features are also continued for the common progenitor, myeloid progenitor. It also has euchromatins, it also has a large nucleus, and it also has a basic cytoplasm, right? Now after that, in the myeloid erythroid progenitor also you will have the chromatin, but the amount of euchromatin will get less because the metabolic activity is slowly decreasing, because it is getting more and more specialized, it is getting more and more differentiated. As you keep on getting more and more differentiated, you require less and less amount of metabolic activity. Now, but the cytoplasm is still basophilic, right? Because a lot of proteins synthesis is going on, transcription rates has reduced. Now we are coming to proerythroblast. So why this is known as a proerythroblast? Proerythroblast. Why proerythroblast? Because this, by now we are very sure that it will not go into megakaryote or megakaryocyte and go into the platelet synthesis. It will from now on definitely go into red blood cell synthesis. That's why we are giving the name proerythroblast. Pro means precursor, right, or primordial form. So it also has, if you look into it very closely, you will see that it also has a basophilic cytoplasm. It also has a nucleus which is a prominent nucleolus. A prominent nucleolus you will find when you have to make a lot of ribosomes. When you have to make a lot of ribosomes, when you require to do a lot of translation, in that situation, you will require a lot of ribosomes, and ribosomes will come from the nucleolus. So this prominent nucleolus shows that in this phase of the development, it is doing a lot of protein synthesis, that is translation. Transcription part is already done in the previous phases, most of the transcription. Over here also some amount of transcription goes on. It's not like that no transcription goes on. Some amount of chromatin is still there, but majorly it does the translation. And as it is doing a translation, it has a lot of mRNA in the cytoplasm. So cytoplasm is still basophilic. I hope this clarifies my point. Now let us move to the next phase. What is the next phase? In the next phase, we have the early normoblast. Now in early normoblast, what happens in this early normoblast phase? Okay, before we come to early normoblast, let us remember one very important thing, that is hemoglobin synthesis. Hemoglobin is a protein, the globin part is a protein. So hemoglobin synthesis starts at the level of proerythroblast, right? We talked about protein synthesis, so this is a very important point that we should remember. After that, what happens? Now we get into the cell stage of early normoblast. Now what happens in the early normoblast? In the early normoblast, as we are getting done with the protein nuclear function, like we don't need that amount of transcription, that is DNA replication and RNA synthesis, because cells are not dividing that much like stem cells do. The cells are not producing a lot of RNA because there is already a lot of RNA in the cytoplasm which is undergoing translation. So the nucleus shrinks in size. Nucleus shrinks in size, but the cytoplasm still remains basophilic. Why? Because a lot of basophilic cytoplasm, because a lot of protein synthesis is going on, and still see we have a prominent nucleolus because it is still producing ribosomes, so that we can have a proper translation going on. After that, over here also a hemoglobin synthesis is going on, so there will be certain patches of hemoglobin to some extent, small, right? Now in the intermediate normoblast phase, the cell becomes visibly loaded with hemoglobin. So the places which are visibly loaded with hemoglobin, those places become eosinophilic, right? Hemoglobin binds to acid. Always remember, hemoglobin binds to acid. So hemoglobin binds to eosinophilic dyes. So it will bind to eosinophilic dyes, right? So as it binds to eosinophilic dyes, the cell remains somewhat eosinophilic, somewhat basophilic. So this kind of a feature is known as polychromasia, multiple colored thing. So polychromasia, that is seen in intermediate normoblast, right? A lot of colors are seen over here. Okay, now over here, first you can visibly notice the hemoglobins. And this presence of hemoglobins gives a eosinophilic appearance. It was already basophilic, so basophilia and hemoglobin eosinophilia together form a polychromasia, multiple colors. Then we move on to the late normoblast. Now over here also see the nucleus has shrunk in size because the functionality of nucleus is decreasing with time. There's no much of function because we are not doing a lot of transcription or DNA synthesis. Now we come to something known as the late normoblast. So what happens in the late normoblast? Late normoblast, you can see that the staining is more or less eosinophilic. The reddish color has appeared because the cytoplasm has got loaded with plenty of hemoglobin, because the hemoglobin synthesis is still going on. Plenty of hemoglobin is present in the cytoplasm, and the nucleus has shrunken even more. More nucleus shrinking. More nuclear shrinking, right? So after that, what happens after this phase? What happens? The nucleus is thrown out, and the cell becomes something known as reticulocyte, which lacks the nucleus. Now why it is known as a reticulocyte? Let us understand. Over here we have a clear cytoplasm which is reddish in color, eosinophilic, because of a lot of hemoglobin is produced present in the cytoplasm. At that point of time there is still some RNA, mRNA, tRNA, rRNA left, because the remnant or the residual part of the hemoglobin synthesis is still going on. So over here you will see some basophilic strands. Over here in the cytoplasm you will see a lot of basophilic strands, which are essentially the RNA which are left behind, which are still undergoing some amount of translation or stuff. So that gives a net like appearance, a basophilic net on the eosinophilic cytoplasm. So that gives a reticulum like appearance. That's why it is known as a reticulocyte. And now see, as we go on from towards maturation, it has assumed the name cyte. That means it is a differentiated. Now it will not modify any further. Later on what happens? The RNAs get resolved, they do not exist anymore in the cytoplasm, and the cell shape modifies into biconcave shape, and finally we get our RBC, right? So this was the detailed discussion on the erythrocyte synthesis. Now few trends I would like you to remember. What are these trends? Let us see. First of all, you can see one thing, that is, as you move from these cells to over here, what happens? This one trend, that is, cell size decreases. Cell size, as you move from hematopoietic stem cell to all the way into the RBC, you will see that gradually the cell size decreases. So that is one very important feature. Another thing that you can notice, that is, the nucleus shrinks. Whenever you have a metabolically very active cell, you will have three features: large nucleus, euchromatin, and basophilic cytoplasm. As the cells become more and more differentiated, they become metabolically less and less active, and less active cells have small nucleus, eosinophilic cytoplasm, and you have small size. Small eosinophilic cytoplasm, small nucleus with heterochromatin. So these are the features, right? So nucleus shrinks, the cell size decreases, the differential, they become more and more differentiated, the differentiation increases. Now over here, the most important points that you should remember always for your exams, that hemoglobin synthesis starts in the proerythroblast. That's very important for your exams. And another thing, nucleus is excluded, nucleus is kicked out at the late normoblast phase, right? Now which phase enters the circulation? The reticulocyte may remain in the bone marrow, or it may remain in the circulation. It remains, in fact it remains in the circulation. 1 to 2% of the entire RBC count that we get in the investigations is reticulocyte, and that reticulocyte gets specialized, it changes its shape due to certain membrane proteins, and that leads to formation of RBC. Now what are the specific membrane proteins we will be discussing later on, while we will be dealing with something known as RBC membrane biochemistry, right, or RBC membrane structure. So I believe by now you're very much clear with the idea of erythropoiesis. Now what we learned in this video, we learned the different cell stages of the erythropoiesis. We learned how the different cells modify, how the eosinophilicity or basophilicity of the cells change, and how from hematopoietic stem cell finally you get an RBC, right? Now one thing before we finish it off that we should remember always, that is, over here let us talk about the early normoblast, intermediate normoblast, and late normoblast. In the early normoblast you can see it is evidently basophilic, so it is also known as basophilic normoblast. Right? Intermediate normoblast, it has a tinge of red as well as blue, so it is known as polychromatic normoblast. The late normoblast has predominantly red. So predominantly red, it is known as orthochromic. Ortho means straight. You can straight away say that it is red or eosinophilic. So it is orthochromic normoblast, also called. Right. So these names, that is your basophilic normoblast, polychromatic normoblast, and orthochromic normoblast are also important. These are asked in examination sometimes. Now, another thing that you should see, that whenever we are moving in the direction of differentiation, what indicates differentiation, just see that from the name blast, we finally moved into the name site. So from blast to site, this also indicates that we are moving towards the direction of increased differentiation, where cells are more and more specialized and more differentiated. Now with this, we finish off this video. If you enjoyed this video, please hit the like button, share this video among your friends in medical school and college. And if you haven't subscribed to my channel by any chance by now, please do hit the subscribe button and press the bell icon and put the option to all, so that you never miss an update from my channel. Until then, bye bye. See you in the next one.